Hello friends, welcome to the Biological Life Sciences channel. Today in this video we are going to see about the details of electrocardiogram, that is also in short form written as ECG or EKG. So let's begin. So electrocardiogram, it's a diagnostic tool that is used generally for the determination of improper functioning of heart. Okay, so generally electrical impulses and muscular function of heart is assessed by this technique through different angles, and different electrodes are attached to the whole body, and then the electrical signals are checked for different parts of the body from the heart, and from heart to different part of the body. Okay, so this is a technique where you can determine the proper functioning of heart. Okay, so generally ECG test, it's a painless and harmless test, and there is a specific machine, that is ECG machine, that records all the electrical impulses all throughout your body, and it does not put electricity in your body, but it calculates the electricity that is present in your body. Okay, so let's see the brief history. First of all, William Einthoven, he is a Dutch physicist. In 1960 to 1927, he made the first ECG recording in 1895. And he also recorded the waves of the heart, that is PQRS waves also was first time defined by William Einthoven in 1895. So in 1905, William he recorded the ECG in his laboratory for a patient who was situated 1.5 kilometer away in the hospital. Okay, so first published his recordings in a different journal. This was the photograph of the first ECG machine that he took. You can see here, it's a old string galvanometer electrograph showing the big machine with the patient running his extremities in the cylindrical electrode. So the extremities such as hands and legs has to be kept in a electrode solution like this, and then the readings were taken. So in 1924, William Einthoven, he received a Nobel Prize for the invention of ECG, and it was indeed a very important discovery. Nowadays also, whatever the problem related to heart, the first and foremost test that is done is ECG. So hence ECG is a very important test, and there is no alternative for ECG till now. So Goldhammer and Scariff were the first to suggest the use of exercise ECG for the diagnosis of coronary artery disease in 1932. Then after that, Charles Wolfert and Francis Wood were the first to report the precordial electrodes. So before this, the electrodes were, as you've seen in the last photograph, the electrodes were immersed, or the solution was immersed into containers, and you have to dip your extremities in such containers. But here in 1932, these two scientists first time reported the use of electrode as a corded electrodes. Okay, so it was quite convenient after that. And in 1942, Emil Goldberg added the augmented limb leads, that is AVR, AVL and AVF, to the Einthoven's 3-lead electrodes. So the first electrode that Einthoven selected was a 3-lead electrode, and it was further developed by Emil Goldberg, and he added the 12-lead electrocardiogram that we see nowadays. So let's first of all, before beginning with the details of the heart, first of all let's see with the basic anatomy of heart. The heart is 4-chambered. The human heart is 4-chambered heart, you know this. There are right and left atrium and right and left ventricle. So the right side of the heart collects the blood from the body and pumps it to the lung for the first circulation, while the left side of the heart receives the blood from lungs and then it pumps it to the whole body. Okay, so blood flows through the heart in this way through 2 times. So the oxygen rich blood from lungs comes from left atrium and through pulmonary vein. Okay, so this circulation, we know it's called as a double circulation. Okay, so the blood then flows into the left ventricle, where it is pumped into aorta and is distributed to the whole body for the metabolism. Okay, so this is how actually the whole heart works. So this is the diagram of double circulation. You can see here, once the blood goes from deoxygenated blood goes from heart to the lungs, and then it is oxygenated here and it comes back to the left atrium, and through the aorta it is distributed to the whole body, and from whole body it again comes back to the right atrium, So likewise the cycle continues, and hence the circulation in heart is called as double circulation. Okay. So let's see the conduction of heart. First of all, the right atrium is a very important site because there is a muscular junction here in the right atrium, right at this place. If I have to show you, there is a muscular junction that is called as SA node. It is also called as sino-auricular node. Okay. Then after that there is a AV node which is situated in ventricle, and then right bundle and left bundle, means right and left bundle here in the heart, and then the electrical impulse spreads through the bundle of His and then through the Purkinje's fiber. Okay. So first of all, here at this position in heart there is a presence of SA node, and then there is a presence of AV node here in this position, and here at the center there is bundle of His, and then it relates to the all the Purkinje's fibers that are spread throughout the ventricle. Okay. So these are the different muscles that helps in conduction of heart. The heart has its own automatic pacemaker. As I told you, the impulse starts from the SA node, that is sino-auricular node. It is located in the right atrium. I just showed you the position, and it is independent of brain, and it generates the electricity for heart. So normally the impulse is generated by SA node, and it runs through the heart's electrical signals and the muscle cells of the atria to beat simultaneously, and allowing for the squeezing of heart and contraction of the heart, and pushes the blood into the ventricles. The electrical signals that are generated by SA node, then it travels to the junction box between arteria and ventricle, where the AV node lies, where it is delayed for few milliseconds to allow the ventricles to fill. So now here the ventricles are going to be filled with blood, and after that it is going to contract. So the signal from SA node is transferred to the arterioventricular node. Now this ventricular node, AV node, this pumps the blood and it goes to the either to the lungs or to the whole body. Okay, so there is a short pause here for allowing the blood to be returning to the heart, and then the cycle repeats itself. So the whole cycle here is shown here in this PQRS form. PR interval, then is QRS complex, then ST, and then T interval, QT interval. Okay, so consequent RR interval or QT intervals are monitored generally in ECGs to check out the rhythm of ECG. So let's see what all these waves has to tell us. Okay, so let's see about the P waves first. The P wave represents arterial depolarization. In sinus rhythm, there should be P waves preceding each QRS complex. So before each QRS complex, there must be a P waves that shows us arterial depolarization. So after that, there is a PR interval. The PR interval is from the start of P waves to the start of Q waves, and it is represented by the time taken for electrical activity to move between arteria and ventricles. Then after that there is a QRS complex. This whole QRS complex wave that is shown represents the depolarization of ventricle. So it can be seen as a three closely related waves on ECG, that is QRS waves. And then the last ST segment, it starts at the end of S waves and finishes at the start of T waves. The ST segment, it is an isoelectric line that represents the time between depolarization and repolarization of the ventricle, that is contraction. Okay, and then comes the T waves. This T waves represents ventricular repolarization, and it is seen as a small waves that is occurring after the QRS complex. Okay, so these are the different waves that are present in the ECG readings. There are correlation between two different waves. The first correlation is RR interval. So the two consequent RR interval is generally monitored. The RR interval, it starts at the peak of one R wave and ends at the peak of next R wave, and it represents the time between QRS complex. Then after that there is QT interval. The QT interval, it starts at the beginning of QRS complex and finishes at T waves. So it represents the time taken for the ventricles to depolarize and then repolarize. Okay, so these are the different functions that this each wave tells you, and what are the condition of the heart, and in that respect is shown to you here in this. Okay, so now let's see The lead that is present here. So the lead is the imaginary line between the two ECG electrodes that is taken as a central axis, and the electrical activity of this lead is measured and recorded as a part of ECG. So generally nowadays 12 lead ECG records are taken, 12 electrodes are there, and then 12 of these leads are producing 12 separate graphs on the ECG paper, and however 10 physical electrodes can be attached to the patient. Okay. The paper in which the paper is also a special one here, which on which the ECG is recorded. So we are going to see the papers also here, paper, graph paper. So the paper in which ECG is recorded is standardized across all hospitals. So the standard paper is there, graph paper having the standard readings. So here each small squares that is present in this paper is represented by 0.04 seconds, and the large squares present is represented by 0.2 seconds. So when 5 large squares are connected together, it therefore represents 1 second, and 300 large squares connected together, it represents 1 minute. Okay. So this is how the reading is taken. You can see the graph paper shown over here. This is the PQRST wave. This is the actual whole function of heart recorded in this waves. So you can see the smallest one is 1 mm here, and the bigger square is 5 mm, and the much bigger square than this is 1 millivolt is shown here. The time intervals is also shown here. You can see this square, it counts for 0.2 seconds, and then the bigger one, it counts for 0.4 seconds, and the largest one counts for 1 second. Okay. So this is how the readings are taken, and the heartbeat is measured, or heart rate is measured. The paper speed which the machine takes, it is about 25 mm per second. So 25 mm of paper is pushed into the printing machine per second. So this should be the speed for recording the heartbeat, and all heartbeats should be same for a proper functioning of heart. If there is an irregular pattern that is observed in this waves, you can see here, then different clinical conditions can be correlated with it. So here there are different leads present which are slightly different in shape. This is due to the lead recording the electrical impulses or activities from different directions. So therefore whenever there is an electrical activity, there is a positive deflection, and if the electrical signal goes away from the heart, then it is negative deflection. Okay. So likewise the deflections are recorded, and the activity is measured, and the wave is created on ECG paper. The height of deflection also represents the amount of electrical signals that is flowing, and it is measured in different directions. Here the R wave is greater than the S wave. It suggests that depolarization is moving towards the lead, and it is recorded in that waveform. And S is greater than R. It suggests that the depolarization is moving away from the lead. Okay. So the R and S are equal size. It means that the depolarization is traveling at exactly 90 degree to the lead. Okay. So this is the calculations or the readings that are being taken by the different leads that results in the different graphs. Okay. So this is the segments that are important here, that is taken by lead 3, that tells you the different segments, and the regularity of each interval is also calculated here. So it all, as I told you, it all starts with the SA node, and then it spreads to the arterioventricular node, that is AV node. Then after that it spreads down to the bundle of Fish, and from there it goes for Purkinje's fiber, which causes the ventricular contraction. Okay. So therefore all these readings are calculated using the different leads here. 4 of the leads are attached to the limbs that is present in 2 legs, and the 2 of the leads are attached. 2 of the leads are in legs, and 2 of the leads are present in hands, extremities of hands, and then 6 leads are placed in the heart, close to heart, and then all the functions of the heart are monitored here, and the graph paper is recorded, and then the reading is made in such a way that you can determine whether there is an irregularity in the heartbeat or heart rhythm that occurs here. So this is how the ECG taken. I hope you have understood the details of ECG and how it is recorded, and what are the waves, that is PQRS waves in ECG tells you. Thank you so much for watching the video. If you like the video, please press like button, share, comment, and subscribe the channel, and Do comment if any queries are there. Thanks for watching.